students. In today's session, let us discuss the structure of mitochondria. So far, we have covered endomembranous system, which includes endoplasmic reticulum, Golgi complex, lysosomes and the vacuole. This session is important as it carries 5 marks weightage. Generally, mitochondria are referred as powerhouse of the cell. It appears like discoidal organ, oval in shape. A section passing through mitochondria will show different components which are present within. Let us begin with its cavity. Cavity of mitochondria is filled with a semi-solid mass known as matrix. In the matrix, they have their own DNA molecules which contain the information transcribed on RNA molecules, so that RNA molecules, they can synthesize their own proteins to regulate mitochondrial reactions. This matrix is surrounded by 2 membranes. Outer membrane is continuous. Inner membrane is showing infoldings, and those infoldings which are suspended in the matrix known as cristae. Between outer and inner membrane, there is a space present called as perimitochondrial space. On the inner surface of cristae, these are minute particles known as elementary particles, Racker's particles, F0, F1 particles or Fernandez-Moron particles. These are various names given. Each elementary or Racker's particle is said to be a reaction center where electron transport system is present. When electrons travel through this electron transport system, the energy is liberated, and this energy is used for phosphorylation of ADP to ATP. So ATP generation is always in mitochondria, hence we call it as powerhouse of the cell. So each mitochondria is having 2 membranes, outer, inner with perimitochondrial space, inner membrane with infoldings called cristae, and when you magnify single cristae, we can see these elementary particles or Racker's particles, and when you magnify Racker particle, it is having the base, stalk and the head region. In this head region, complex 4 is present. In stalk region, complex 3 is present. In base, complex 1 and complex 2 are present. In the unit 4, under the topic respiration in plants, we are learning the electron transport system of Racker's particle known for production of ATP. It is known as oxidative phosphorylation. In that topic, we are covering the actual mechanism of ATP production. So here, I'll just explain you in brief. Whenever cell utilizes a molecule like carbohydrate, that is C6H12O6, now stepwise it is metabolized into carbon dioxide and water, liberating stepwise hydrogen. And for this liberation of hydrogen, this process we call oxidation, the cell utilizes molecular oxygen. This we call cellular oxidation of the substrate. Here substrate taken is glucose molecule. It is a carbohydrate. So when a substrate glucose, which is an energy producer, undergo oxidation in presence of oxygen, liberating hydrogen, giving end product carbon dioxide and water. Now what will happen to these hydrogens? These are carried by the carriers NAD, FAD. So when they reacting with the hydrogen, they are reduced to NADH2 and FADH2. Now in complex 1 of Racker's particle, NADH2 enters. It gives H2 and oxidized as NAD, while here complex 2 is present. This is entry for FADH2. It releases hydrogen and oxidizes to FAD. So NADH2 oxidized to NAD. Releasing hydrogen is in complex 1, while FADH2 undergo oxidation in complex 2, releasing hydrogen and oxidizing to FAD. Now these hydrogen molecules will split, giving 2H plus protons outside and liberates electrons. So H2 splits into protons and electrons. Now let us move from complex 1. These electrons are accepted by complex 3. When a pair of electron will move from complex 1 to 2, 2 to 3 and 3 to 4 present in the head. During this electron transfer, the cytochrome system which is available inside, it takes the radiant energy from the electron and the escaped energy received by ADP, it is adenosine diphosphate. It stores that energy in this bond by reacting with another phosphate and generates ATP. So, a pair of electron transferred from complex 1 to 2 with the liberation of energy which is used for phosphorylation of ADP to ATP. Now a pair of electron which is it is with the complex 2 moving on complex 3 again liberates energy. So that energy used by ADP and undergo phosphorylation to generate ATP. Now the electrons are with complex 3 moving to complex 4. Again energy is liberated, accepted by ADP reacting with phosphate to form ATP. So here electron pair transferred from complex 1 to 2, 2 to 3, 3 to 4, generating first ATP, second ATP and third ATP. So NADH2 after entering in mitochondria moving through Racker's particle yields 3 ATP molecules. Now already I told you FADH2 reduced here. Hydrogen is with complex 2. Undergo dissociation liberating protons and electrons. A pair of electron moving from complex 2 to 3 here. Now this phase gone. From 2 to 3 giving first ATP, 3 to 4 giving second ATP. So when FADH2 carry hydrogen during cellular oxidation to mitochondria, going in the cristae, entering in the base of Racker's particle, received by complex 2 yielding 2 ATP molecules. This we call terminal oxidation occurring in aerobic respiration. Now if you think prokaryotic cell and eukaryotic cell, in prokaryotes mitochondria are absent. So energy yield is very less compared to eukaryotic cells, and in eukaryotic cells mitochondria are freely suspended in the cytoplasm having numerous Racker's particles utilizing hydrogen and generating ATP molecules. That's why mitochondria is referred as powerhouse of the cell, because this mitochondria will help in terminal oxidation, and terminal oxidation occurs in Racker's particle. Okay, so matrix of mitochondria and elementary particle of mitochondria helping in complete oxidation of substrate into CO2 and H2O. With this, I'll conclude the session on mitochondria. Thank you very much.